All right, so to start Unit 4, we are going to talk about, in general, cell communication. Then we're going to kind of skip around to a lot of different chapters that have specific examples of how cells communicate with one another. And then we'll kind of end on the cell cycle and regulation. Kind of strange, but that's how they lumped Unit 4 together. So here we go. This slide just kind of covers the general pieces of what you need to know. There's 3 parts of cell communication: reception, transduction, and response, and we'll go through those. And just how the receptor part actually recognizes a signal and what does that mean, and usually there's all kinds of signaling inside the cell that happens, and there's different ways to do it. There's phosphorylation cascades that we will learn. There's also something called a second messenger. And basically, how does the cell turn genes on, turn things off, and then a lot of downstream responses, for example, in the cytoplasm, turn things on and off. And then at the very end, we'll talk about why apoptosis is important and not just a bad thing. So, cell signaling. Animal cells communicate by direct contact. So they are physically connected to one another via something called gap junctions that we'll talk about. Animal cells also communicate by doing what's called local signaling. So this is when one cell secretes some kind of signaling molecule to a very close surrounding environment. This is done by growth factors, as you can imagine, are designed to help something grow, and neurotransmitters, which we will talk about when we get to the nervous system and synapses and brain cells and things like that. There's also long distance signaling. So how can something from the brain, for example, target your adrenal glands that sit on top of your kidneys? Those are usually done by the endocrine system via hormones. So there's lots of different levels of signaling, whether it's physically connected in the close surrounding area or even long distances across the body. Here's a couple examples of what that looks like. The top is gap junctions between animal cells, so you can kind of see what that looks like. Plant cells have something very similar, but they're not called gap junctions. They're called plasmodesmata, and it's basically the cell wall is connected with passageways for things to move. Cell-to-cell recognition, this would be a local regulator where the antigens or presented proteins on one cell are being recognized by some kind of receptor on another cell, and when that lock key kind of fits, then that triggers a reaction in the receiving cell. Here's a picture taken from your book on kind of what the local and long distance signaling is. We had mentioned already, kind of B here, that synaptic signaling or neuron signaling is one example of local. You have one nerve cell here releasing a bunch of neurotransmitters or signals that will then cross over and connect to a target cell that has a whole bunch of receptors on it. And those receptors are going to receive the signal, and then an action is going to result. Paracrine signaling is also another local signaling thing. It is, again, where secretory vesicles are unloading some kind of growth factor or signaling molecule that will then do something similar and go along to receptors on a close cell in the surrounding area and have a response. The long distance signaling are the hormonal signaling or endocrine system. So this is where some kind of endocrine cell, whether it's in your pituitary or your hypothalamus or your thyroid or your adrenal glands, wherever it may be, your pancreas for insulin is going to release hormones that can then travel through the bloodstream, and if they're in their bloodstream, they can pretty much go anywhere. And so they will act upon target cells by binding the hormone specifically. Okay, so now we're to the 3 stages of cell signaling, and this is kind of a generalistic picture of what's happening when parts of your body are trying to communicate to each other. The plasma membrane plays a huge role in this. So this is tying it back to the idea that the proteins in the plasma membrane can act as receptors. They can receive signals. And that's where this is going to play a role, because then when they receive signals, then there are downstream or later consequences inside the cell as a result. The first thing that happens in cell signaling is called reception, and that's where the cell detects some kind of signaling molecule. It's usually called a ligand. Some people say ligand, I say ligand, whatever. But that is the usual name given to a signaling molecule. And oftentimes it's coming from outside the cell, whether it's from a very far away cell or a cell from the close community of cells that it's living in. Some kind of signal is just kind of floating out there, signaling molecule ligand, and it will be able to be detected by a target cell. That is reception. It's received the signal. Then comes transduction. That is where that signal, once recognized, is converted to something the cell can actually use or actually has a response to. So that conversion oftentimes is kind of like a lock and a key situation. The ligand will bind to some kind of protein on the surface of the cell and oftentimes cause a conformational change. And when that happens, that triggers a whole bunch of later action steps. And that's really what the transduction is. It's transducing or converting the signal into something that actually triggers a response inside the cell. And then the last piece is the actual response. It's the response to the signal molecule. Once that message has been received and transduced, then the actual cell can respond. Here's what that generally looks like. On the left, you can see that this signaling molecule or ligand comes and actually binds to a protein on the membrane. In this case, it's a receptor that has a unique fit, right, to that ligand. And when that happens, there's some kind of transformation of shape, for example, and there's all these downstream changes, and then at the end there's some kind of response. In this case, we're turning something on, you're activating it. So the binding of this ligand to the outside of the cell triggers some kind of activation inside the cell. It could also be inhibition, but in this case it's activation. Okay, so in more detail, reception. Go through all these and then kind of, talk about it. Binding between the signal, which is the ligand, and the receptor is highly specific. The membrane protein is not an enzyme, but it's pretty much similar to how an enzyme acts, right? Like there's a lock and a key or induced fit model of an enzyme where they come together and something happens. It's a similar idea. A ligand will fit to a specific receptor and they are highly specific. So acetylcholine, for example, is a neurotransmitter that will only bind to acetylcholine receptors. What's another one? Epinephrine will only bind to epinephrine receptors. And there's different kinds of receptors. I've kind of been talking about the plasma membrane receptors, the ones that are really inside the membrane itself, but they don't have to be. There are also intracellular receptors. These are the ones inside the cell. And so those are for small hydrophobic ligand molecules. They can actually make their way through the membrane on their own. So they don't have the membrane as a barrier. So they go inside the cell to reach the receptor target. Or they're located inside the cell because the things that attach to it don't need to remain outside. So testosterone is an example. Nitric oxide is an example of two ligands that will come straight through the membrane and attach to these intracellular receptors. So long story short, if the ligand molecule can actually make it through the membrane, then it can get to receptors that are inside. If the ligand has properties that would prevent it from normally crossing the membrane, well, then its receptors are on the surface. So it's going to bind to the receptor protein. There's going to be some kind of shape change as a result. And then that is what triggers the transduction signal, some kind of conversion or several downstream steps. Okay, one example of this whole type of receptor is called a G protein coupled receptor. It will get abbreviated GPCR, and there are thousands of them. They are extremely diverse, but they all generally function in the same way. You have this kind of multiple transmembrane protein. So it's showing you the overall structure of how it's folded. If you remember way back when we learned about primary, secondary, tertiary, quaternary structures. Alpha helices were secondary, and then we have a tertiary kind of 3D folding here. But the idea is it goes transmembrane, and more often than not, it crosses exactly 7 times. So you can see that it zigzags 1, 2, 3, 4, 5, 6, 7. One end is outside the cell, one end is inside the cell. There's a special pocket where the signal will bind. So this is the special area where the ligand will bind. And then down here is a specific segment that's going to act with something called a G protein. And I'll talk about it in a minute. But what usually happens is a ligand binds up here, and that will cause a little change, and then it's going to react within the cell down here. You can see a kind of more 3D picture of what that looks like on the right. Here's it showing roughly how it works. So if a signaling molecule binds, that kind of activates the G protein coupled receptor, the GPCR. And this involves an energy molecule or a G protein called GTP. It's very, very similar to ATP. ATP is adenosine triphosphate. GTP is guanine triphosphate. Similar type deal. But that signaling molecule, when it attaches, activates essentially, or fuels up, it powers the G protein. And that's why it's called G protein coupled, because that little protein basically just hangs out there unactivated. And when a signaling molecule finally comes along, then GTP fuels it up. And then it can come over here to an inactive enzyme, whether it's in the membrane or not in the membrane. But it kind of comes over here and turns it on. It activates it in some way. So the activated G protein leaves the receptor, travels over this way, and activates the enzyme. Well, once you have an activated enzyme, now it can actually do something. So that's where the cellular response comes in. All right, so here's another example. This one is called ligand-gated ion channels. So it's a little bit different. In this case, a ligand is coming and it will bind to, instead of a G protein coupled receptor, you can see there's no G protein that's stuck onto it. It's really a channel that will open or close in response to something. And those ion channels, when they're open, will allow ions to flow through. And when it's closed, it won't. So normally what happens is the channel is closed and there's something called a ligand-gated ion channel receptor. So it's still a receptor, you still have a ligand binding to it, it's just normally closed. And this is usually embedded in the membrane. So then when the ligand binds to it, it causes a conformational change and that gate opens. And when that gate opens, a whole bunch of ions can either kind of go in or go out. In this case, they're going in. And because of that, that will trigger a cellular response. When the ligand unbinds, then it essentially closes the gate. So the response in this case is not to an activation via a G protein, it's activation because of the ions that have moved from one side of the membrane to the other. Okay, so intracellular receptors. Too far. Here we go. They can be inside the cytoplasm, or they can even be in the nucleus. Remember, the ligands for these are just coming straight through the plasma membrane, so they are usually hydrophobic in nature. They are the ones that are able to get across the intermembrane hydrophobic tail area. And so these include Nitric oxide, remember, steroid hormones, thyroid hormones. And as we will learn, when those signals or ligands actually bind to the intracellular receptor, wherever it may be, it becomes activated. And it can act as something called a transcription factor. Transcription, you may remember from biology, but that's where you turn certain genes on. You take the DNA and you turn it into RNA. And so if it's a transcription factor, that means that it has the ability to control what genes get turned on and off. So that's very powerful because it's a steroid hormone, for example, when it binds to a receptor, can now turn certain genes on or turn certain genes off, which is why hormones can have drastic effects. Here's a picture of just what that might look like. So there's a hormone up top, and it can make its way naturally through the plasma membrane because of its hydrophobic nature. It binds to the receptor protein, and it's intracellular because it's inside the cytoplasm. Once it becomes activated, in this case, it then goes to the nucleus, which makes sense because if it's going to control transcription, it needs to get to the DNA, and the DNA lives in the nucleus. So it goes in here and attaches, and then it can control, in this case, it's turning something on. It turns on this gene, and then it's going to create a new protein that wasn't being made before. So here's a table that kind of goes through the different types of receptors. On the left, we had the G protein coupled, which again had the 7 transmembrane segments, and there's a partner G protein that becomes activated and can trigger a response by activating some kind of enzyme to do something. Intracellular proteins are the ones that are inside, and the activated complex can turn genes on and off. And then lastly, the ligand-gated ion channels. Those are the ones where a ligand binds to the channel and then oftentimes opens it up to allow the flow of ions. And then that flow triggers some kind of an immune response, or not immune, some kind of signaling response. Okay, so this brings us to the second step, transduction. This takes a signal that was originally received and actually turns it into something that's usable, which brings up a couple of key vocabulary words. A cascade is just a very general way of saying a bunch of interactions that eventually get to your target molecule. So it's kind of like a series of dominoes that gets you from the initial signal to the eventual target. It's a cascade. This oftentimes involves protein kinases. A kinase is something that goes along and activates a protein. It has ACE in it, so it is an enzyme, and it phosphorylates it. So it tacks on a phosphate, and by doing that, it activates the protein. So in this series of dominoes, or this what will become known as a phosphorylation cascade, it kind of, one will enhance the next, enhance the next, enhance the next. And this is done by a series of phosphorylations. Generally speaking, when something becomes phosphorylated, that means that it's activated. And so if this is happening down a cascade or a signaling cascade, then it's activating everything necessary to get to that eventual target. Here's what it might look like in a picture form. So where do those phosphates actually come from? For phosphorylation, ATP. It's dropping off one of those phosphates and turning into ADP. So in this case, the ligand binds to the surface receptor, and that will then activate some kind of molecule. And an inactive protein kinase now becomes activated. So this could be a G-coupled protein receptor, for example. And what happens is the next enzyme comes along and phosphorylates it, and now makes it active because it has a little phosphorylation. And then you get an inactive protein turning into an active protein. So they kind of just act in this continual phosphorylating way, and the proteins can be kind of reused or recycled. So the inactivated protein gets phosphorylated and then transfers it to a specific protein and then recycles. Does it again and again and again. You can also have a second messenger. A second messenger is similar or a part of G-coupled protein receptors, but it's not as direct. So in this case, some kind of molecule or ion is going to relay the signal inside. The most general one that's used often is called cyclic AMP or CAMP, however you want to call it. Cyclic AMP is kind of one of the hallmark second messengers. So one example, I guess, in the bottom here is using calcium, which could be another second messenger. This happens a lot in signaling too. But you get the activated, the ligand binds. That's kind of the first messenger, right? Like it's the first signaling molecule binding to the receptor. Then you have your special activated G protein that activates an enzyme to go and do something. So here, this activated enzyme actually triggers this second messenger called IP3 to go into the endoplasmic reticulum, and it kind of can act as a ligand because it's binding to a calcium-gated ion channel. So it can act as a messenger that opens up the ion channel, and then the calcium goes off and does something. So this is a very long-winded idea, but it's not just the initial ligand that's doing signaling. There are other molecules that are acting as a second messenger down the line to carry on the signal to other parts of the cell. Cyclic AMP literally stands for cyclic adenosine monophosphate. There's also, in the process, we go from a G-coupled protein receptor to something called adenyl cyclase, which is the enzyme that needs to be activated. And it can turn ATP into cyclic AMP. And it, disregard my children in the background. ATP can turn into cyclic AMP that can act as a second messenger to do things later on. So again, energy is still required, but in a little bit different way. Which leads us to the response. So in this point, the response means that the cell is actually going to do something once the signal has finally made its way to the endpoint. And this could be turning certain genes on and off, like in this picture. There's some kind of growth factor acting as a ligand. It triggers the receptor. In this case, there's a phosphorylation cascade. So things are getting phosphorylated, moving down the line, using up ATP. The signal molecule will make its way into the nucleus, and then that will then trigger the response of turning on a gene. It's regulating protein synthesis, and therefore it can regulate the activity of different proteins. So it can either make more of a protein, kind of amplify a response, or it can also turn off the production of a protein by inhibiting a response. So that's really how these initial signals, when they get to a cell, eventually trigger some kind of on-off response. There is a thing you can go to if you want to kind of see what that looks like in action. Okay, so now we're getting to some various examples of signal transduction pathways. When we get to the test, there's going to be a lot of different example-based problems of how this can apply. So diabetes is one that we'll talk about again when we get to endocrine. Cholera, there's immune diseases, there's cancer, there's neurotoxin. And pretty much the reason why now we're going to go through different types of signaling like endocrine, like immune, like neuron, are all because they involve signal transduction pathways, cells communicating to cells, and a lot of things can go wrong with that. So cholera is one example. It's contracted by drinking contaminated water. It's caused by a specific bacteria that likes to live in your small intestine, and it makes a toxin. And why is it so bad? The toxin actually modifies the G protein. So the G protein that sticks to the receptor in G protein coupled receptors, at least the specific one that's involved in salt and water secretion, is modified. And what happens is it sticks to the active form, and it causes the intestinal cells to secrete water, which is not great, because if you are secreting water 100% of the time, you are having profuse diarrhea. And the idea is cholera just basically makes you lose all of your fluids too quickly to replenish, and you essentially die of dehydration. Also, we have something that's related via Viagra, and I'm sure you've all heard of this before, because it is used in the treatment of erectile dysfunction. Fun fact, it was all initially discovered as a blood pressure medication, and then they found out that it had this sudden side effect. But what does it do? How does it work? It inhibits hydrolysis, or the breaking down, of cyclic GMP, which is very similar to cyclic AMP, down to GMP. And what does this do? It prolongs the signal to relax smooth muscle in the artery walls. Overall, it's increasing blood flow to the penis, therefore treating erectile dysfunction. It's kind of acting as an inhibitory ligand molecule to have this end result. But you can also see why it was originally proposed as a blood pressure medication, because it increases blood flow. Here's what that generally looks like. You have your brain, and it's making a whole bunch of stuff. Inevitably, what's happening is in this molecule, in this flow, the breakdown of cyclic GMP in green to GMP in blue is inhibited. So therefore, you're going to get stuck on the increased blood flow path. Okay, last but not least, we're going to talk about cell suicide. So in cell suicide, that's apoptosis. It is also known as programmed cell death, which is how I prefer to look at it. The cell is broken down and digested, and this is triggered by something called caspases. It's an ACE, so it's an enzyme, and caspases are what are called suicide proteins. They are signaling molecules that trigger the death of the cell. Why would a cell do this? Well, to protect neighboring cells. So kind of a cell can monitor itself, and when it realizes that it's hurt or damaged or broken in some way, it will get rid of its own self before it damages the cells around it. But what's important to note is that this is very important in normal development and maintenance. So for example, in your hand, you have different fingers. We don't have webbed hands. If we didn't have apoptosis, we would have webbed hands. Apoptosis actually causes the removal of all of those webbed portions in between our fingers so that we actually have fingers. So it is part of normal development as well. It's also thought to be involved in certain diseases where particular brain cells are more prone to death than others. So this result references Parkinson's, for example, or Alzheimer's disease. You can kind of see what apoptosis looks like. On the left, you have a normal white blood cell, and on the right, you can see it's starting to shrink and form little blebs. That's a hallmark sign that it is no longer alive. And here's kind of what I was mentioning in terms of your fingers. So this is a mouse paw, and mice have limbs too. On the left, you can see before apoptosis, and on the middle, you can kind of see the particular cells where apoptosis is happening. And then on the right, lo and behold, you end up with actual digits that a person might have. So that's just one specific application. But that kind of concludes overall cell signaling. In the next couple presentations, we'll go through specific examples where these signaling types are used. So you're going to have to be able to take the general idea and apply it to a variety of situations.